This video is going to focus on hormones and their actions and how they're transported through the blood. And Dr. Lin's original PowerPoint that he put in there, he kind of skipped this, and I put this slide back in. So if you're following along on your slideshow, you don't have this slide. But his first slide used these words. So I thought it might be helpful to actually classify these hormones. But I can also understand why he skipped this because it's pretty detailed. So hormones can be broken up into three chemical classes. Again, we call them chemical messengers. That just means that they're compounds, and they can fall into these three chemical classes. So steroids, which are derived from cholesterol, monoamines, which are made from amino acids, and then peptides or glycoproteins. And those are chains of amino acids. And I included that picture right next to it just so you can kind of see. I know that might not mean a whole lot to you, but the steroid is in the blue, and then the amine, monoamine, is in the pink. You know, the amine group just means it has an NH2. And then the peptides are the yellow, and so they can be a short peptide chain or a longer chain, as in insulin. So these words are going to be used coming up, talking about how hormones transport. And so I thought it would be easier to understand what those categories mean. But I'm not going to test you over. I'm not going to show you a picture of one of these and tell me, is this a peptide? I don't need you to memorize any chemical formulas here. I just wanted to show you what the differences were. And keep in mind that one of those monoamines is very similar to one of those yellow dots on the peptide. So amino acid and then a chain of amino acids is very similar. So how are these transported through the blood? And again, I think it helps to understand those classifications a little bit before we dive into this. So most monoamines and peptides are what we call hydrophilic, which means they are water-loving and they mix easily with the blood plasma. Basically, they can just dive right in, okay? But then not all hormones are that way. So other hormones, especially steroids and thyroid hormone, are hydrophobic, which means they do not mix well with water. And basically they need to bind to someone to help them through this watery blood that they are traveling through. So they bind to transport proteins. Some examples are albumins and globulins. And these transport proteins basically carry the hormone through the blood. It's called bound hormones if they are basically riding along on a transport protein, or an unbound hormone if they are by themselves. And so a bound hormone that's riding on this transport protein, it has a longer half-life, which basically means it's protected. It doesn't break apart. The protein is protecting it from breaking apart, and it has a longer life. Whereas hormones that are unbound, they don't have that protection, so they're not usually lasting as long in the blood. Only unbound hormones are able to leave the capillaries to reach the target cell. So the hormone eventually does have to break off from the transport protein when it's ready to actually get to where it's going and go to the target cell. I'm not really picky that you memorize these three. Thyroid hormone has three transport proteins that it rides along with: albumin, thyroidine, and thyroxine binding globulin. But more than 99% of thyroid hormone is protein bound, so that means it's riding on a transport protein. Steroid hormones usually bind to globulins. Aldosterone has a short half-life. That's kind of the time that it can ride around in the blood. And so 85% of that hormone is unbound, and only 15% binds to a protein. Again, I don't need you to memorize that. I just want you to see that when it binds to the protein, it stays along a lot longer. We're going to find out that thyroid hormones stay a long time. That's going to be an example later. Okay, so once the hormones have made their way through the blood, they are kind of reaching their target cell. Well, how do they know when they're there? Hormones stimulate only those cells that have receptors for them. So depending on what type of cell it is, they have different receptors. Now, you're probably comfortable with receptors back from muscle cells and neurons, but receptors are protein or glycoprotein molecules. They're on the plasma membrane usually. Now, with some hormones, they can be in the cytoplasm or even in the nucleus, but we're really comfortable with them being on the membrane. And they're kind of like switches turning on a pathway when the hormone binds to them. That just means there's a reaction ready to get started, but it's waiting for the hormone to get there before the reaction takes place. So sometimes usually a target will have a few thousand receptors for a given hormone. And these interactions are specific, kind of like an enzyme is specific to its reaction that it helps. They're very specific receptors for each hormone, and they also can be saturated. That just means when all of the receptor molecules are occupied, then that's it. They're kind of all full. Okay, then this goes into basically the difference of once they get there between hydrophobic and hydrophilic hormones. So hydrophobic hormones are able to move through the cell membrane and enter the nucleus, and they're going to be able to act directly on the genes. And when they do that, they are targeting a certain type of cell, the physiology of that type of cell, so they have a specific function. When they are nuclear receptors, that means the receptor in the nucleus, then that can take longer to show the effect. And that's because of protein synthesis needing to occur before the effect is noticed. Whereas hydrophilic hormones, they cannot penetrate the target cell, they cannot get inside, and so they stimulate the physiology indirectly. Now, that first part makes sense to me. That's no problem. But when you start talking about indirectly and then you start talking about second messengers, it gets a little fuzzy. And so before I flip to this next slide, I want you to realize that I know that this is getting a little fuzzy. A lot fuzzy. So here I just want you to not get too wound up about this and just know that when we're talking about hydrophilic hormones, they have to basically stimulate a second messenger activation or a second messenger. So basically, this is the way I think about it. They're knocking at the door, they're telling someone they're there, but they can't go in. So somebody inside has to go get the reaction started. So it's kind of like when your kid is at the door and they're not allowed to let you in and okay, go get your mom. Okay, I will. And so that's how I can remember it. But I don't want you to get too wound up about this process. I'm not going to test you over this process. But just realize that this camp is kind of that second messenger. Okay, so not only is the hormone reaching the target cell, but as we just saw in that last example, that they can trigger a cascade of reactions. So they are extremely potent chemicals. So one hormone molecule is not just triggering one reaction molecule. It's bigger than that. So one hormone molecule can trigger the synthesis of many enzyme molecules. Your book gives the example of one hormone making 1,000 CAMP molecules, okay, like in the previous slide. And so if then 1,000 of those CAMP molecules make 1,000 enzymes each, okay, we'll think about 1,000 of the CAMP molecules making each 1,000 enzymes, okay, well then those 1,000 enzymes are going to eventually make, and this is kind of a exponential process, but you're at 1 billion molecules of the product, whatever that product may be. And that's all from one hormone molecule. So a very small stimulus can make a big effect, and the circulating concentrations are very low, but then within the target cell, that's when we start to see this amplification. Target cells can be adjusted by changing the number of receptors. So kind of how sensitive they are to the hormone can change based on their number of receptors. And so if we get more receptors or a cell increases the number of receptors, it's called upregulation. And then the opposite would be downregulation if they reduce the number of receptors. Upregulation example is in late pregnancy, the uterus produces oxytocin receptors, kind of getting ready for the surge of oxytocin that it expects during childbirth. That would be an example of upregulation. And then downregulation, decreasing, especially if target cells seem to have a constant long-term exposure to a hormone. They seem to reduce the number of receptors and become less sensitive. Okay, this last little part of this is about hormone interactions. We would expect different hormones to be targeting different cells, but most cells are sensitive to more than one hormone. And when those hormones, more than one hormone, basically are there at the same time, we can see these different interactions. So you can have a synergistic effect where more than one hormone act together. You could have a permissive effect where one hormone kind of primes the target and gets them ready for the second hormone to come later. Or antagonistic, which means the hormones are opposing each other, such as insulin and glycogen. And then the last final piece of this hormone puzzle is that hormone signals have to be turned off once they've served their purpose. So earlier we used the phrase half-life, and that just means the time that the hormone is circulating in the blood. And when that half-life is up, or when that time is over, most hormones are taken up and degraded by the liver and the kidney, and they are excreted in the bile or the urine. And so that gives you a metabolic clearance rate. That's the rate of hormone removal from the blood. And so the half-life is the time required to clear half of that hormone from the blood. And so the faster that clearance rate, the shorter the half-life of the hormone.